Bismillahirrahmanirrahim. In the name of Allah, the most gracious, the most merciful. It is a common knowledge that maggots are considered as a nuisance. They are associated with garbage and spoiled food, and to an extent, may assist in diabetic patients. Nonetheless, today I would like to bring a perspective that these creatures can play a significant role for forensic death investigations. Here, the title of my presentation is When the Maggot Whispers, the Overlooked Clues in Forensic Death Investigations. Before I go any further, I would like to express gratitude to the organizer of the International Conference on Sharia Law and Science 2024 and the University Science Islam Malaysia for inviting me to share the little knowledge that Allah has bestowed onto me in forensic sciences. My name is NAJI ARAFAT bin Haji MAHAT, a senior lecturer in forensic science from University Technology Malaysia. Ladies and gentlemen, this picture shows a decomposing body that has been largely infested by maggots. When a forensic investigator faces such presumably a murder case, there are several pertinent scientific and legal questions that need to be answered in order to complete the investigation. Firstly, how long has the victim been dead? This is particularly a challenging question since at this stage of decomposition, the common body changes, namely the algor mortis or the reduced body temperature, at this stage, the body temperature may be similar to that of empyrean. Meanwhile, rigor mortis or stiffening of muscle and livor mortis or lividity have long been gone and no longer informative for investigation. Secondly, what could be the probable cause of death? Was he stabbed or poisoned? Can we analyze the rotting tissues for poison at this stage of decomposition? Wouldn't the poison be already degraded and tissues are not suitable for analysis at this time of decomposition? Thirdly, who is this victim? Can we still identify by the conventional means of DNA and fingerprint? Fourthly, was it a foul play? Was this a suicide case? Or is it a homicide or accidental? Next, has the body been altered after death by the perpetrator? Did the perpetrator spray the body with insecticide in his or her attempt to delay the coming of flies, to confuse the forensic entomologist in estimating the post-mortem interval so that he or she can have an alibi of a few hours or a day? Lastly, is this the real primary crime scene where the victim was murdered? Is it possible that the victim was killed elsewhere and the body was dumped here? In this context, would you believe me if I said that maggot may whisper us the answers? Ladies and gentlemen, now let us dive further in understanding the basic principle for using forensic entomological evidence in investigating death cases. Studies have shown that beyond 72 hours after death, pathological changes like algor mortis, livor mortis, and rigor mortis would not offer much clue for estimating the PMI or post-mortem interval. In this regard, utilization of entomological specimens, chiefly the necrophagous insect, has been suggested. The fundamental of its utilization relates to the fact that insects are poikilotherms or cold-blooded, which means they are unable to generate body heat for sustaining the biochemical and physiological processes required to sustain their lives. They depend on external heat for that matter. Therefore, its developmental pattern remains predictable as a factor of heat unit, also known as the accumulated degree days or accumulated degree hours. Well, this slide number 4, it looks like a newspaper, but this is the fake one. It's nothing real about this. I just made it for the purpose of the presentation anyway. Now, the question is, how do maggots help in solving death investigations? The answer is maggots provide us with four important clues or facts, namely, by providing accurate estimation of PMI, possible post-mortem relocation of a body, the identity of the deceased, as well as the possible cause of death and circumstances surrounding deaths. Now let us scrutinize one by one. PMI can be estimated via two approaches. Firstly, when the body is recently dead. For example, in 3 days to 2 weeks, PMI can be estimated using the developmental pattern of the oldest necrophagous species. In Peninsular Malaysia, the species are of Chrysomia megacephala and Chrysomia uvivaces. Perhaps the distribution can be different from the other part of the country like Sabah and Sarawak. Secondly, whenever the body exceeded 2 weeks or so, the PMI can be estimated by interpreting the species composition, more like a snapshot matrix kind of interpretation. For example, species A, B, and C are found during week 1. B, C, and D during week 2. C, D, and E during week 3, and so on and so forth. This approach requires accurate taxonomic identification of genus and species, as well as understanding on the species composition of the location. Next, I will explain about post-mortem relocation of the body and human identification together by giving an example. For example, out of anger, Ali was murdered by Ahmad in an office on Saturday. After the murder, Ahmad went home. At home, Ahmad was very anxious that he had murdered someone and left Ali's body in the office. Ahmad was afraid that somebody or someone may discover Ali's body on Monday because it's going to be a working day. Because of this, Ahmad went to the office the next day, which is Sunday, with the intention to move Ali's body to somewhere else, so that hoping that he will not get caught. Ahmad then wrapped Ali's body with a rug and put the body in his car trunk. Ahmad then drove to the outskirt area and dumped the poor Ali's body in a primary jungle. After 1 day, the police discovered a body upon receiving a report from a hunter nearby. During the post-mortem examination, the pathologist sampled the infesting maggots, and the species was ascertained by a forensic entomologist as Synthesiomyia nudicita. You don't have to remember the name anyway, right? So a common fly that is found indoor near human establishment. The forensic entomologist went on explaining that it was impossible to find this species on a body killed in the jungle. As a matter of fact, if the body was killed in the jungle, the species infesting the body should have been Chrysomia villanuvae, or what we call as the jungle dweller. At the very same time, Ali's absence had caused confusion at the office. A janitor found maggots and trace of blood at the office and subsequently made a police report. The samples of maggot were collected by the police, and the same forensic entomologist later identified them as Synthesiomyia nudicita. That is, the same species found on the body from the primary jungle. That is suspicious, right? This puzzled the forensic entomologist. He then asked the investigator to send the samples for human DNA analysis from the gut content of the maggot. Upon performing the DNA analysis of the body, as well as the gut content of the maggot, it is established that the two profiles belonged to the same person, which was Ali. Therefore, the analysis of entomological evidence has proven the fact that Ali was killed in the office and dumped in the jungle, proving the fact that the post-mortem relocation of Ali's body from the primary crime scene to the secondary one. Finally, the entomological evidence can also suggest the possible cause of death and even its circumstances surrounding death. For example, if a person wanted to commit suicide, he or she may want to choose a poison that is readily available in the market, like pesticides such as malathion and paraquat, and then gulp the poison. And of course, to commit suicide, he or she will choose a remote or secluded area that is far away from people so that no one would interrupt the attempt to commit suicide. Because of that, the body may not be found for a few days. People may realize the odor of decay and make a police report after a few days. When discovered, the body may be in the advanced stages of decomposition and liquefaction has taken place. At this particular stage of decomposition, the body tissue may not be possible to be analyzed, considering the advanced stage of decomposition. However, maggots are on the body and relatively they are fresh. Therefore, the maggots can be analyzed for poisons and or drug. In this context, even the qualitative detection of malathion or paraquat is sufficient to suggest the possible cause of death. This is because malathion and paraquat are poisons that can readily cause death, and no one ever abused malathion and paraquat for recreational purposes. Unlike these poisons, drugs like morphine and cocaine are not readily causing death. These drugs are abused and people would not necessarily die due to the consumption of these drugs. So if we analyze the maggots from the body that contain morphine and cocaine, we may not be able to suggest the cause of death. To the very best, we can suggest the circumstances surrounding that the person abused these drugs during his lifetime. Still an indication for investigation. Now, are we clear on how maggots are useful in death investigation by providing us these 4 important facts to the investigation? Who says that maggots are only nuisance? Every creature that Allah has created has its own role and function. Now, let me introduce to you the maggot of Chrysomia megacephala. This slide 5 depicts the photomicrographs of the salient trait for us to identify the third instar larvae of Chrysomia megacephala. In this slide number 6, it shows the salient traits for identifying the adult Chrysomia megacephala. As you can see, the conspicuously large eye facets, right, and the gena and postgenal areas are entirely orange-yellow and clothed with pale yellow hairs, prothoracic sparkle that are dark, and at least the lower squama distinctly infuscated. Now we are moving towards the end of my presentation. In slide 7, it would not be complete if I do not discuss about challenges and the way forward in forensic entomology. I categorize them into scientific and non-scientific challenges. Firstly, the continuous introduction of drugs and poison in the market that can potentially affect the necrophagous insects. We know that there are drugs and poisons that can alter the life cycle of necrophagous insects, which in turn affect the accuracy of the PMI estimate should their presence are not factored in the estimation of the post-mortem interval. Since new drugs and poisons are entering the market on a daily basis, concerted efforts are required to perform forensic entomological studies that look into the effect of these drugs on necrophagous insects, as well as developing the specific analytical methods for analyzing these drugs and poisons. Secondly, Baseline data developed for one biogeoclimatic region may not be useful for others. Considering that insects' composition and behavior differ from one region to another, there is a need to perform forensic entomological studies in varying regions as well as seasons. Thirdly, taxonomic identification can be challenging. Necrophagous insects, especially the maggots, may look similar to the untrained eyes, especially among the Sarcophagidae. Since the accuracy of PMI estimate relies largely on accurate taxonomic identification, the unavailability of well-trained taxonomies may hinder the investigation. Fourthly, the study of interkingdom interactions of necrobiomes and necrophagous insects is now gaining the attention of forensic entomologists in Western countries. They theorize that the arrival and colonization of necrophagous insects are affected by the chemoattractants produced by necrobiomes on the decomposing body. This aspect is also referred to as quorum sensing. Despite such a rapid development in this aspect, the same in Malaysia remains nascent, whereby this aspect has not been the focal research area by the Malaysian forensic entomologists. The challenges are not only on scientific ground, there are also challenges that are related to the non-scientific aspect. The inadequate training by the investigation personnel relating to the importance of entomological evidence, the proper procedure of collection and analysis. To overcome this issue, continuous training shall be provided to them in identifying the possible exhibits to be collected, how to preserve and rear the living maggots before transporting the specimen to the forensic entomologist for analysis. Another challenge is the fear to testify in court, limiting the number of practicing forensic entomologists. This could be one of the deterrents for the ordinary entomologist to venture into being a forensic expert. This can be addressed by having specific court appearance training so that the novice forensic entomologist can be educated in terms of how to appropriately deliver their testimonies and answer the questions by lawyers in the court of law. Guys, that's all about it. That would be the end of my presentation today. Thank you for listening, and once again, I would like to express my gratitude to the organizing committee for the opportunity given to me today. Thank you very much for listening. Assalamualaikum warahmatullahi wabarakatuh.